see. This is Anne. Could I ask a question? I have a comment and a question, if I might. Whoops.
Go ahead.
Sorry, I'm having trouble with my with my machines here. A comment and then a question. Dennis, this beautiful talk, and you mentioned if we're not going to have seizures as an outcome, what are we going to have? And you have to have something else, and maybe we'll have people who don't have seizures. And I just want to emphasize that in some of these disorders, seizures are not the main thing. And probably one of the poster childs for that right now is KCNQ2 DEE, where children's seizures often resolve, often very early on, but they're left with horrific neurodevelopmental disorders. And in fact, when I worked with the KCNQ2 community, they said, oh, put the seizure questionnaire at the back. That's not our big problem. What they really wanted to focus on was sleep and constipation, and then behavior. Those are their biggest concerns. So a trial for KCNQ2 in older children is not going to be about seizures for the most part. The other thing I wanted to mention, you had talked about the delayed start design to demonstrate disease modification properties of a treatment. And that's a real hard one. And I wonder, you know, in the infantile spasms trial literature and the NCL, the DMD, the SMA, we don't require that. We know that if a drug is started earlier, it stops the progression of the disease. And if it's started later, it will stop the progression of the disease, but it doesn't return someone to optimal health. So how much does the FDA actually need to see people randomized based on stage of disease?
Yeah, that could be FDA hardball, because the FDA can talk tough and then can back off sometimes, not always. I think the FDA is concerned about claims of disease modification. The term is thrown around, I think, too much. I think by the agency's standards. So some of that may just be a warning sign of, you know what? Yeah, we all want to do better trials and we want the patients to be better in many ways, but be careful about that term. So yeah, whether they mean that, I don't know, but that's a fair point.
Thanks.
There's been some questions about a preclinical. Well, Anne, you asked one of them. You want to speak up? I see you just popped up.
I did. I would love to. We're talking about trying to have more mechanistic trials for LGS, and whether that's LGS as a group or specific subsets. And I think when we're faced with the question of, is this something worth doing? Is this something where the potential risks are going to be mitigated by potential benefits from our patients? We really do want to know, what do the models show? What data are there to suggest that there's a real possibility of benefit? And so I guess I just wanted to start a discussion on what are the features that this group would like to see in terms of preclinical features you might see in an animal model that could in some way, if not exactly replicate, in some way present as surrogates for the clinical features in the children who actually, those features should really matter. This should be the features we want to target. So apart from seizures, which I think we agree would be important, what are the other things that animal models should be, or cellular models should be trying to recapitulate and showing that there's effect from some of the preclinical attempts at trials?
Tracy, I just want to jump in, but should we raise that question tomorrow after the preclinical talks and merge everything from today with tomorrow? I mean, or we can try to answer that now, but
you answer it now, or then everybody for tomorrow can kind
of. No. I mean, I'm not going to answer it now. I don't have an answer. Sorry, I didn't mean it that way. Let someone else answer it now. But
go ahead, Eric.
No. I was just. What's your thought on that? Do you know?
Yeah, I think it'd be nice to cover that in more detail tomorrow. But I think people can certainly give their opinion. But a lot of what I'm hearing is there's this heterogeneity, and yet we all converge onto the same EEG patterns, even though those might be difficult to read, right? Even though you might have inter reader reliability that is not perfect, right? We all converge, no matter where we came from, onto these EEG features. And there seems to be, that we're learning that even though there is this spectrum, there are certain things about those EEG features that suggest a network level dysfunction. So I think if you're studying LGS, those would be the things you want to do. But there's a whole spectrum of pre-LGS that we're not talking about here, where you can target the specific genes, or you could target full body cooling in infants with hypoxic ischemia to try to prevent the evolution to LGS. Or you could target the 3 areas that Dr. Greenspan mentioned, right? If you wanted to do high dose steroids in this gap period, or any of these other pieces, or if you could stop it at spasms. And so I think, you know, just given the nature of this discussion, thinking about how we could target people who already have LGS, but not leaving out that spectrum. There's a ton of our advocacy groups are already trying to address those early issues, and we want to support them, not compete with them. But what if you don't respond to an SCN2A gene therapy, right? Or a Dravet therapy, or brain cooling, or full body cooling, and you do evolve. And then I think that's where we want to start our first levels of research. But I think tomorrow is a good place to cover, but I'm certainly, if anybody knows the answer to that question, by all means.
I mean, I put my answer in the chat to Dr. Paduri's question, but I can answer verbally as well. You know, at our place, we've been struggling with exactly those issues. You know, there's we had a bench neuroscientist who came to us and said, I think I figured out Monk 18.1. And we're like, what is that? She's like, oh, it's STXBP1, and I know what that is because that's a cause of, you know, of epilepsy and epileptic encephalopathy and other problems. And we looked at what she did in the dish, and one of them was an FDA-approved drug, phenylbutyrate. So and it looks to be a safe drug that they've already approved, and you know, for urea cycle disorders. So we very rapidly worked to get a trial together to test the safety and efficacy of that in this narrow population. The idea of steroids have come up. One of my clinical colleagues, it's her practice. Like, someone with LGS has really bad epilepsy, she brings them in, she gives them 5 days of high-dose IV steroids, or she gives them these high-dose dexamethasone regimens. And you know, we published our findings based on what she's done with her patients and some of our own. But that idea has never been tested in a clinical trial. It's you know, IV steroids are an old medication. We just need to figure out how to test whether it works based on these sort of anecdotal case series kind of interventions. So I think there's something incredibly attractive and amazing about really diving in, understanding you know, at a very fine level, what's the disease causing biology. But I think we need to also take a look at, well, what are people doing in practice? What seems to work, to try to unpack if there are successful strategies already in use that could help us optimize care. You know, with this, and I'm thinking about our comparative effectiveness with Levator, Acetaminophen, and Phenobarbital. We had the ideas like, oh, let's see what people are using for early life epilepsy. Keppra, you know, Levator, Acetaminophen, and Phenobarbital were the number 1 and 2 in use, and we compared them, and the effect was dramatic. That Levator, Acetaminophen, it was like 40% efficacious versus 16% for Phenobarbital. You know, with the outcome measure that we used in this comparative effectiveness design. So there may be a lot of stuff that people are doing already that doesn't rest on a lab. That doesn't rest on the bench neuroscience and the translational neuroscience. But I suspect that there's a spectrum, that there's going to be kind of low-hanging fruit where there are things that look safe and effective, we can just jump on it. And then there's going to be stuff that like is really getting in and modifying the disease at its core, but has potential for safety and tolerability problems that we need to be more gentle about. That was a little bit of a tirade. I hope that was okay.
John, you have your hand up.
Yeah, so I was just going to make a couple of comments. The first is just to highlight an interesting finding of this Estelle paper, which hopefully will get out, be out very soon. But we also did overnight EEGs, ambulatory EEGs on these patients, and quantified the burden of paroxysmal fast activity. And in particular, if you look at the longer bursts, 5 seconds or longer, and you add up how much of that's occurred in a 2-hour window of sleep, it strongly predicted the frequency of seizures over the next 1 to 3 months. So it looks like certainly the more prolonged bursts of PFA are going to be a very useful biomarker of treatment effect. And I think that then becomes, an important thing for shortening the duration of trials. You can look at treatments that reduce the burden of those and then have fairly high confidence it's going to be effective in your subsequent trial. So that's the first thing I wanted to say. The other thing, I'm a strong believer that the field of the discharges is very important. And I personally, when I see an EEG going past, which has got sustained trains of essentially centrotemporal spikes or spike activity over the centrotemporal regions, even if it's relentless and occupying the whole record, I think that has a very different impact on the brain than discharges that have a more sort of typical frontocentrally dominant spike wave pattern. And my sense is you even see that in the cognitive, we had a child even last week hammering away from the pericentral cortex, cognition relatively good. The different seizure activity in different networks has different impacts. So personally, I think there's perhaps, I like to see more attention paid to the field of the discharges, because I think that obviously will impact what network is being involved and hence what cognitive impact is occurring.
John, just to follow up that, one of the issues which I think Dennis raised before, right, is that some of these findings can be state dependent, can be time dependent, age dependent, right? So, someone can have slow spike wave for 6 months and then never have it again. So to hone in on these EEG features and looking really specifically, you start to potentially have, biases based upon when and how you're looking. So how would you suggest to think about that and take that into account when you're really diving into the EEG?
I think I think the danger is if you just include anyone with a sort of sharp, slow-looking thing on their EEG, is that you're really just looking at bad epilepsy and you're not really getting down to which networks are really driving it. And then that starts to contaminate any effect you're going to have on treatment. If you want to have a treatment that's targeting the relevant network, you've got to start to zone in on what are the core features that define that. I agree that different networks are sensitive at different ages. I mean, that's why relandic epilepsy happens at the age it does, presumably. So, I think the features of LGS reflect the network being involved, and then the different sharps, PFA and slow spike wave reflect the mode of failure within those networks. Again, personally, I think PFA is a core feature because it looks similar to the start of tonic seizures, and I think it's really saying something very bad is happening to generate those bursts of fast activity. Not saying that slow spike wave can't lead into that, and that's also a marker that it's perhaps heading down that path, but it's the PFA that's really telling you the problem.
There's been lots of questions going on here, but going also just online, there's been a few that I don't want to leave those people out since they've been entering questions. Just to get into, for Jennifer, there's a few questions about your experiments in sleep versus wake, and if you do consistent intervention, do you permanently reverse things?
So I think we're still working on this. I don't want to speak too much definitively because we don't have all of the conclusive results of our study back sufficiently powered yet. In terms of the state dependence, that's an important question. And so in some of our previous work that I mentioned involving the memory tasks, we were looking specifically at sleep after learning. So we were really trying to hone in on an epoch of time and a state that would be important for memory consolidation. So there can be a role for state-dependent interventions, such as during sleep, that could potentially have ramifications beyond just that state. Now, the closed-loop experiments that we're doing right now, we're doing state-independent therapy. So basically we're going through wakefulness and sleep as well. And in some of our rats, we actually have a little bit surprisingly seen a reversal of the epileptic process. So not only does the cortex look better, but the hippocampus starts to look better as well, which we don't have a good explanation for yet, but seems interesting. So yes, that's definitely where we're focusing our investigation to try and understand that better.
Thank you. Then just also from the people who are streaming, someone asked for Dr. Dlugos, how do you get all these different specialists and these people who work on these different rare diseases to communicate better in order to improve the outcomes and improve the way that we do clinical trials? What are your thoughts on looking outside of the LGS community?
Well, the issues are different. There are some unique issues for each rare syndrome, but then there are some common themes. So, there are, options to collaborate. The Rare Disease Network is one. The Research Roundtable for Epilepsy is another. The Epilepsy Foundation of America is another. I mean, the collaboration is important, but you're still going to be left with what are the unique features of your condition that are robust enough to use as an outcome measure. I don't want to minimize the value of collaboration, but it's not. There are still, each condition is different. But probably the Epilepsy Foundation, which has a research arm, is one opportunity to do that. But yeah, that's it's a challenge. Can I also address some? There was a line in a thread in the chat about EEGs and LG. The inclusion criteria for LGS trials are typically by design broad as possible to fit under the LGS umbrella. They have LGS, but very broadly defined. So I don't disagree with some of the comments about we need to focus on patients with certain EEG features or active EEG features. The reality of that is many patients will not be eligible, and the studies get longer and more expensive and more onerous for families. Like, bring us your current slow spike wave or PFA burden, as opposed to an EEG report from a year ago that says an epoch of slow spike and wave you're in. So those are good, but those are different from the current bevy of studies, and they are more onerous for families and more patients don't qualify. So that's just the reality of being more, trying to be more specific.
But be more rigorous, maybe.
Yeah.
I don't think that's necessarily a bad thing, Dennis. I mean, I think it's just different because right now, at a certain point, how many drugs for LGS that have all been designed approximately the same way but haven't had a main impact are going to, is the market going to bear, right? We try everything. We try everything. We try palliative brain surgery. So adding the burden of getting a repeat EEG for the chance at a different type of treatment might be appealing to many families who are struggling with intense seizure burden. But it also would close the gap. But I think at a certain point, the world may say, gosh, we've made all these LGS drugs, we don't want to keep doing it, or they may continue. So we have to think about other avenues for the future.
Mm-hmm. Yep.
And then just circling back to a few older questions, I think it will stir up the discussion a little bit more. But during Brenda asked about the connection between your nodes of the GPI and the cerebellum, and I think this also then feeds into John's different cortically based networks and brainstem thalamic interactions. How do you guys think those during yours was on TSC versus LGS? Do you think these networks are shared across them, or they're really separate based upon disease? And as you know, patients with TSC develop LGS. So where do you think the overlap comes between them and the separation of them?
I'm not entirely sure, and I think the first order of business is to look at lesional LGS. That's why I'm happy that Dr. Archer is on the call as well, because I know he has a number of lesional cases sitting in his on his disk drive somewhere. But if you look at the EEG signatures and even the clinical semiology of tonic seizures and spasm, there's so much overlap that I would imagine that there's network properties that are shared. I like the idea of testing hypotheses with multiple modalities, like what Dr. Archer has been doing. So repeating the same finding in different disorders would be great. It would be even better to confirm this in another way. The GPI is connected intimately to many other structures, so maybe not a very specific finding, but it is where, in this case, a lot of things came together. One way to refine it also is to see if we can reduce the number of candidate lesions that are actually relevant, because there may be a lot of lesions sitting on this network but not necessarily disrupting it. And I think that those that actively prod the network with epileptic discharges are the guilty ones. So I'm not 100% sure yet what I'm doing with 2 nodes as opposed to 1. I don't have a brainstem node, but I don't know if my normative connectome actually entailed the brainstem connections. I don't think it did, and not low enough maybe to look at similar pathways. So there's a lot of work to be done. This is kind of my first venture out into the spasms network on an imaging level.
John, do you have any thoughts on that?
First comment is we should definitely have a chat about calibration, and I read Jurian's paper about the location of lesions, and I thought that was very interesting. It's very parallel to thoughts we've been having for a while about plotting the location of lesions on the brain, because I really do get a sense that there's certain brain regions don't cause it. I mean, why is it that you don't get LGS from hippocampal sclerosis? Why does LGS almost never have hippocampal sclerosis? Why don't they have lesions in the pericentral cortex? You start looking at the key sort of PFA activation map, and you go, well, that you can start to see why. It's just not those brain networks. Even though they're being hammered with seizures, they don't get HS. It's not mesial temporal, limbic epilepsy. It's just a different beast. So I fully agree with you, the importance of networks. I'm not sure about the GPI. I in spasms, I see your data and it's convincing. I suppose my question is about a sort of a shared exit path being seen, rather than necessarily being a key driver, and how you tease that apart. It might be. I don't know. I think it'd be interesting to sort that out. My guess before seeing your work was that spasms was the cortex down to brainstem and out without going back up through the thalamus part. And as the brain matures, you start cycling up through the thalamus and coordinating the cortical activity. Because at the end of the day, spasms don't have a scalp EEG signature. Why not? Why do they just get the big fat slow wave and not much else going on? Clearly widespread cortex is not being activated, and yet clearly it's driven from the cortex, because you chop out a cortical lesion and it goes away. So my sense is that it's, again, this very primitive alerting startle network that you see. That often patients with LGS have startle-induced seizures. It's this cortico-reticular pathway is essentially being driven. So that was my guess, but I'd love. We're trying to get funding to do a EEG fMRI study in spasms, but it's obviously got a lot of challenges to it with the age of the children. But Aaron's just put in a grant application for that. It'll be very interesting to see what we see. I don't know if that answers your question at all.
I'm muted. It does, or if this is all food for thought, because, how do we grapple and how do we get our hands around all these EEG changes and what these networks are? right, to try to think about treatments. And, towards that, Jennifer, Brenda earlier said something about some of her STIM patients that she's done, the central median nucleus, they seem to, the stimulation wears off, or when they turn it back on, they get another response. And then John just asked the question about, duty cycle, continuous stimulation versus responsive stimulation, and then getting towards an preclinical model aspect of this. What would be the ideal stimulation paradigms, and sleep versus wake stimulation, as you already mentioned. Where do you think we can go with this from a preclinical perspective to teach us about what we should be doing in any trials, device-related trials?
Yeah, so I think on a theoretic basis, if we really parse down and try and understand what the electrical stimulation is doing in terms of biasing neural firing, or like biasing the timing of the neural firing, or actually, stimulating neurons to fire or be silent, I think that responsive stimulation that, responds, not necessarily just to a seizure, the way that NeuroPace does, but actually to more specific features of the extracellular signal, could really be beneficial. So there was a paper recently where they were looking at responsive stimulation, actually for seizures, but they delivered the stimulation either in phase with the ongoing repetitive cycling of the ictal discharge, or at random. And they found a very strong effect of the phase stimulation compared to the random stimulation. And so I think that if we are trying to develop, responsive neurostimulation mechanisms, where we have a specific underlying mechanism of dysfunction, the responsive aspect of the stimulation is going to be critical. And so we're definitely doing experiments to try and look into that by not only delivering just this closed loop stimulation, but delivering the same stimulation, but not yoked to the occurrence of the IEDs. And my gut is that it's not going to work, or it's going to make things worse. But, this is the reason why we do experiments, to see what the actual outcome is. Because certainly it would be easier just to be able to deliver open loop stimulation. But I think that closed loop stimulation may be more effective, and it also may be better for avoiding off-target effects that could, maybe disrupt the physiologic function of the network at other times.
Yeah, it's a very interesting question, and I like your thought of stimulating, sort of moving away perhaps from the epileptic discharges, which in a sense are a biomarker of the network misbehaving, and trying to find some other sort of marker of when the networks are in an epileptic state, and stimulating at that point to prevent the epileptic discharge, rather than chasing your tail the whole time and sort of trying to switch it off whenever you see the discharge. Very good.
So there's been a lot of discussion in the chat here about, in clinical trials and reliability of EEG reading, and ways to approach detection of events, whether it's PFA or slow spike wave. Do any of the speakers have thoughts about how they would approach that? Dr. Douglas just said it would be really expensive and a lot to ask to do, but how would you guys all approach thinking about, level playing field EEG, clinical EEG analysis? I know, Zach, you already raised the Dr. Hussein's spasm type approach, but what are your guys' thoughts on ways to make it such that we're all thinking about or approaching the same thing from an EEG perspective?
So my last comment in the chat was just a reminder that our EEG success stories normalize the abnormality. Absence drugs normalize the 3 Hz spike and wave. ACTH normalizes the hips. EEG is not necessarily normal, but the hips goes away. Sure, somebody has an investigational treatment that can do that. Absolutely, it will be worth the money and the hassle and the cost of recording all those baseline EEGs to see that go away. Reducing it, I don't know. So anyway, that's just, so sure, we have EEG success stories, but they normalize the abnormality.
All right, well, we're running down on time here. And I think one thing to say from today and going into tomorrow, which was Anne and Scott's question from earlier, is that everything we learned from today, what would we take to move forward as the features that we'd want to study in a preclinical model? What are those core components of LGS? Is it the GPFA? Is it slow spike wave? Is it the cognitive dysfunction that would want to be studied on whatever animal model that people are applying it to? And I think that's something that we need to think broadly about as we start as we listen to the talks tomorrow going forward. So I want to thank all the speakers for their incredible jobs and really nice talks that you gave, and the vigorous chat going on there. And sorry we couldn't get to it all. And then Tracy, I'll put it back to you to kind of end the day.
Great, thank you so much. I want to thank Brenda Porter and Eric Marsh for leading such an amazing session. I think that all of those talks were fantastic. I think that, listening to the first session this morning that laid out the spectrum of LGS, what we know about it, we think there's even more. It was heartbreaking to hear, all of the complications and the frustrations with LGS, but there was so much hope among all of the parents and families that were here, that a group of people like you all have come together to try to help us think about how we can do better for the families. And we can't thank you enough. And then in this afternoon session, it was a very sobering discussion about how much hard work we have ahead of us to roll up our sleeves and to figure out where the opportunities lies and where the gaps are. Is it really finding, using specific phenotypic findings, right? Things that our patients do and throwing off-label treatments at them and seeing if that works. Is it targeting the network specifically with a random stimulation or phased, or targeting different nuclei, or is it going into animal models, or is it all of these, right? And so I'm really looking forward to continuing this discussion tomorrow. I think we can continue to make therapies the way we've been making them in LGS, and there's a path to do that. But maybe we want to think too about how in the future we can be making more targeted, more precision therapies for LGS, and then also begin to think about these pre-LGS. So I cannot thank you enough for giving me and many of our families some hope, because you guys are all really brilliant people, and you're here and you care about this topic. And so we're very grateful for that. We're looking forward to continuing the discussion tomorrow. And we have the hard task in the afternoon of trying to come out with something that could somehow evolve into a request for grant applications from the foundation in the future. So I hope you'll all join us. For everybody at home, thank you for watching. The live stream is going to be ending now. For everyone on the Zoom call, thank you again. You are welcome to stay. We're going to stay on for a little bit longer and just debrief a little bit more about the call. And if you asked questions, we'll try to get those answered to you and get those back to you as soon as possible. It's been a pretty robust chat. Thank you so much, everybody, and you guys have a great day.